This is an embryo that's just been fertilized. This is the DNA that was provided by the mother, and this is the DNA that was provided by the father. Basically, what we're going to watch is this embryo developing and all the cells dividing. In the first division, you can see these two spots appearing. These are going to be the two nuclei of the new cells. So now you can see that this is now a two-celled egg. So we can play this recording fast, and we can watch all the cells dividing. At each cell division, the cells replicate their DNA, so they have 2 times as much, and then the cells divide so that each cell gets the right amount. So you get the worm starting to elongate, and then the muscles become active, so it starts twitching around. It starts off as one cell, as we do, and it divides and forms 1,000 cells. And in those 1,000 cells, you have muscles, you have neurons, you have skin, you have intestines, just like we do. The genes control how all of these tissues are made, how the organism develops. There are genes that control the production of muscles and how nerves are made, behavior too. This worm right here is a mutant, and as you can see, it can't move very well. This is what we call an...
To find this and every other gene in the worm, the plan is to read every letter of the nematode's DNA, looking for glimpses of the genes that make and operate it. The organ he chose was the human brain.
Maybe a third of all the human genome is expressed specifically in the human brain. That could mean 30,000 genes are specific for brain function. So it's going to be extremely complicated to work it out. Yet this is what codes for our personality, our mental processes. Everything we do as a human comes from our brain. So understanding that, we thought, was the key to understanding human life.
The search for genes to read begins with frozen human tissue, in this case, frozen human brain. The cells it's made of contain all 6 billion letters needed to spell out the entire human body. Venter's strategy is to read just those letters that make up the genes needed to operate the brain. Reading DNA begins with breaking it up and putting the fragments into microbes that make millions of copies of the DNA as they grow on a laboratory plate.
On the plate here, we have the particular colonies that have the inserts we want. There's blue and white colonies you can see on here. The white colonies are the one that have the particular piece of DNA in them that we're interested in. We know part of the sequence, but we don't know the whole thing. So that's what we're interested in right now, is getting the rest of the sequence done.
For research that is seeking to divine life's secrets and help discover what makes us human, the work itself is repetitive and mechanical. Machines do most of the reading of the letters in the DNA fragments. And computers help the researchers figure out how to assemble the fragments into the long strings of letters that constitute genes. At this level, all life looks the same.
We share the same chemical structure, the same code with every single species that we know about. So from insects, bacteria, yeast, plants, you would not be able to tell just by looking whether this sequence came from a human chromosome or from a plant chromosome or from a fruit fly chromosome.
All living things use the same 4-letter language. But as researchers sift through our DNA, they are finding that we also share with the rest of life many of the same genes. It's as if our brains contain the instructions to make bits and pieces of everything else alive, from flies to elephants.
There's so many different species, all using that same genetic information. We're looking at all of humankind at a totally different level, and we're finding that we're related to every other species on this planet at a level far beyond what most people ever considered possible.
Of all the discoveries being made as we explore the human genome, the realization that we share much of our DNA with other living things is the most profound. It means that we can look in the DNA of other creatures to learn about ourselves. Mysteries is how a human baby grows from a single fertilized egg. As the egg divides, then divides again and again, to form a mass of thousands, then millions of cells, a dance occurs that seems miraculous. Yet today, the genes that control this dance are being discovered and turn out to be the same genes in creatures as different as the nematode, flies, humans, or as here, a chick. The single gene that can make us a boy or a girl has already been found, and the hunt is on for genes that may make us grow old and eventually die. That our DNA can mean the difference between life and death is the motive behind one of the most intensive hunts for a human gene, a gene that can cause breast cancer. It's a search researchers believe is on the brink of success.
If this works, you will have succeeded.